Hello, everybody. We are going to be presenting a new study showing findings about Parkinson's disease in relation to fecal microbiota transplants as a new treatment option for Parkinson's disease. All right, so it might sound a little confusing. We're using gut bacteria to fight Parkinson's disease, which is typically associated with being a neurodegenerative disorder, which the brain, last I checked, is pretty far away from the gut. So there's a lot of questions that people might have here. So we're going to try our best to break it down in a way that makes it very understandable for potential treatment options and just really just informing about this new fascinating connection we found. All right. So to start off, what is Parkinson's disease? A lot of people here in the audience probably know somebody or know of people who have been impacted by Parkinson's disease. It is a rare neurodegenerative disorder, but one of the more common neurodegenerative disorders in the state. And actually, in the state of North Carolina, we have a Parkinson's disease age-adjusted death rate higher than the national average. And one of the reasons why we are speaking to all of y'all here in Charlotte today is because Mecklenburg County that Charlotte is in, has one of the top 25 highest Parkinson's disease death rates. And if you are unfamiliar with anybody who has Parkinson's disease, some notable figures are Michael J. Fox, Ozzy Osbourne, Muhammad Ali, George Bush, Rupert Holmes, among many others. All right. So, where does the gut play into all of this? Kind of as I touched on before. So, the gut and brain are actually connected way more than you might expect. So everybody has a lot of bacteria in their gut. This is stuff that is byproducts of the food that we eat and helps break down the food that we eat. But for the most part, we want that staying inside the gut. When that starts to get outside the gut, that's when stuff gets a little dicey. And this happens, we found actually a lot in people with Parkinson's disease. It actually can a lot of times first manifest through a breakdown of the gut barrier that allows that nasty bacteria that we really just want to stay in the gut to get out of the gut and travel to the brain via something called the brain gut axis, which is like picture like a little highway for this bacteria to go up to the brain. And once in the brain, once it reaches the brain is usually protected by something called the brain blood barrier. But again, because Parkinson's is a neurodegenerative disorder, this is all starting to break down, and bacteria is able to get into the brain and start to affect a part of the brain that is associated with motor functions, which is why Parkinson's disease really starts to present with those loss of motor functions. That's a lot in part due to that harmful bacteria kind of escaping, breaking out of the gut, and getting to the brain. So, this is where the whole concept of a fecal microbiota transplant comes in because Parkinson's disease will initially start out by somebody having a bad gut microbiome, and a fecal microbiota transplant will take fecal matter or poop from a healthy patient, put in someone with Parkinson's disease, that will just help kind of improve that gut microbiome and prevent nasty bacteria from the brain. So they're going to do this through taking 45 laboratory mice. They all were treated in correspondence with various ethics committees. So we have 45 mice, and we're breaking them into three groups. One's a control group. They're not getting any induced Parkinson's disease or Parkinson's disease adjacent things. And there are two experimental groups. One is the experimental control group. They are getting induced Parkinson's disease-like symptoms through a drug called rhodiola, which is a pesticide that has similar effects that mirror that of Parkinson's disease. And the experimental treatment group is getting the same treatment of rhodiola, but also getting a fecal microbiota transplant. And they're just going to be monitoring the effects of how it will affect them. So these three groups, all of them are tested daily to assess motor function. Various tests were performed to assess their gut health, the integrity of their gut wall, their integrity of the brain blood barrier, which is what keeps that bacteria typically out of the brain. They'll also see how their overall skills are doing. They'll analyze brain tissue just through various different tests, assays, and other techniques.
So for the results, we have seven figures to show. The first figure gives a basic rundown of how mice were treated and the different tests that they were performed with, like balance test, tape removal test, grip strength test, and descending a pole. And in these results, we see improvement with fecal microbiota transplant compared to Parkinson's-induced mice. In figure two, We're seeing that in Parkinson's induced mice, neurons that communicate with each other, there's degradation there, which will cause symptoms of Parkinson's disease to develop. In figure three, we see how the blood-brain barrier is degraded and how this relates to Parkinson's disease being induced, reducing motor function. But in FMT mice, we see this function actually restoring itself and recreating motor function within human body and cells. And figure four is just measuring different toxins that different microbiota produce. And they all, in our Parkinson's induced mice, there's increased inflammation, which will cause uh, the degradation of these neurons. And in FMT mice, we see the opposite trend where there's no inflammation response. In figure five, it's the same thing, just pro-inflammatory products that are made. Uh, Parkinson's disease mice increase this. FMT mice show similar levels to our controls, so there's no effect. In figure six is just a rundown of all the microbiota that was discovered in this experiment, and displaying their relative abundance and differentiating between the different microbiotas in our control, Parkinson's disease induced mice and FMT mice. And in figure seven, we see correlations between the Parkinson's disease mice and how their different microbiotas interact and how that's causing correlation of motor neuron degradation. And figure 7G shows the overall correlation between different microorganisms and how each of them performed in the test and what related to each disease.
So what do all of these results mean? Well, first of all, we see that FMT treatment is strongly effective in this experiment with these mice. The two main things that we want to look at that can really be seen in figures one and two are stronger physical performance with FMT and more stable gut health as well. An additional note here on the left, you can see FMT in pill form, which is not what the mice were administered, but this is a viable form of treatment for humans. So as far as the physical performance goes, a few of the graphs in figure one show that the mice treated with FMT displayed better cardio when put on the rotor rod, which is shown in the upper left image. Better manual dexterity in the sticker removal test that they did. Stronger grip strength, which you can see in the bottom picture. And then finally, better mobility on the pole drop test where they had to descend from a pole. Now, primarily shown in figure two, we see that the FMT mice had much better gastrointestinal health. So the FMT improved the rate of defecation within the mice, as well as the pellet count. The water content within the stool was much higher, indicating that things were moving more fluidly throughout the intestines. And then finally, the distance traveled in the colon, measured with essentially a blue dye, was much further. So the stool was making it from point A to point B at a much higher rate.
Conclusion. FMP corrected, restored gut barrier, reduced inflammation, and protected neurons in a mouse model of Parkinson's. This supports the gun brain axis role in PD and suggests FMT as a promising therapy, meaning that for future medical research, this could be a very viable option for people looking to correct their gut health. Future ideas. Conduct human clinical trials to test safety and effectiveness. Develop targeted probiotics or engineered microbes to replicate FMT's benefits. study gut-brain mechanisms with advanced tools, and explore personalized microbiome-based therapies and assess potential for prevention in at-risk individuals. And for AI usage, three of our members did not use any AI to do for any of this assignment at all. Me personally, I use limited AI for brainstorming and for grammar checks and for checking references, and also using AI to help me make sure that everything lined up with our rubric to complete the assignment. And thank you everyone for watching this. Hope you learned something new. And this is our link to the article that we use to conduct this on, so you can check that out as well if you were curious of where we got our research from.